Hi, everyone. We are back after the short break. Let me bring up the session plan for the rest of the day. We are going to start right in the beginning with the talk by Dr. Veronica Miller, who is the director of the Forum for Collaborative Research and a professor at University of California, Berkeley. She is an expert in regulatory science. And I came to know her through another group in our Rare as One Chan Zuckerberg group, and heard a talk from her about like when they analyzed external controls in FDA decision making and what they learned from that. So that's kind of how we ended up inviting her, so that we can learn and from the outsider's perspective as to what actually ends up working in terms of external controls. From her session, we'll move on to a short presentation by Fabrizio, I think, a few slides on what we are proposing for the retrospective natural studies, so that people will have time, can respond to that. And then we'll jump right into the panel discussion, which we will be joined by Nicolai, who's been here with us so far. But also I want to take this moment to introduce Kerry Jolie, who is joining us from the FDA, from the rare disease branch, correct, Kerry? Do I have it right?
Yes. So hi, sorry. So I'm the Associate Director for Rare Diseases, and I lead the Rare Diseases team within the Division of Rare Diseases and Medical Genetics.
Okay, thank you so much. We are glad to have you. From that, we'll move on to a session that Grant Schulert will be leading, kind of thinking about the data sets that we do have in our field, like studies that were done, if we can get nuggets out of that to use as external controls, and kind of wrap up with a working session. So with that, I am going to pass it over to Veronica right now. Thanks, Veronica.
Are you able to see my slides?
Yes.
Good. Okay. So first of all, thank you so much, Rashmi, for inviting me to come here. It's a real honor to be a part of this Next Gen conference. And I want to really congratulate you and your colleagues at the foundation for the amazing work that you are doing, and getting the field together around this very challenging rare disease of systemic juvenile idiopathic arthritis. It's so necessary to have the whole field come together to be able to find treatments for diseases such as this. I just want to make sure that it's clear that the perspective and opinions that I will be presenting here and also during the discussion are my own. I'm not speaking on behalf of any particular group. And I just want to make sure that is clear. I always like to start off with this sort of general framework, because I think if we always keep this in mind, then we can really think about how data fits in. So for drug approval, and drugs here include biologics and of course vaccines and diagnostics as well, it's really all about the benefit and risk. And we talk about that a lot. We want the benefit to be greater than the risk. And that is clear. And sometimes we're willing to accept more risk than other times, but it's really all about the benefit and the risk. But the third part that really comes into here is uncertainty. And to what level of uncertainty are we comfortable living with? And I found Janet Woodcock's commentary in Clinical Trials in 2018 very enlightening. Her first sentence in that commentary is, drug development can be described as the progressive reduction of uncertainty about the effects of a molecule in people. And I use that a lot when I teach my courses at Berkeley. And here I've just tried to kind of represent it. We have at the beginning of drug development, as we go forward in time, there's a lot of uncertainty. And the more studies we do, the more experience we have also in clinical practice. this uncertainty decreases. And at some point, hopefully, we'll have 100% certainty, but I don't think we ever really get to 100% certainty. But at which point are we willing to make a decision? And that will be so dependent on the perspective that we're all coming from. Are we a parent of a child with a rapidly progressing rare disease, or are we a regulator, or are we a payer? And so, again, there's so much in regulatory science that is a matter of opinion and judgment, and that includes benefit and risk and uncertainty. And although we try to develop frameworks that we can apply more and more consistently, in the end, very often there will be different opinions as to what level of benefit and risk and uncertainty we're willing to accept. So when we talk about external controls, we're really talking about data that comes from outside of a randomized clinical trial. And usually we also think about that in terms of real world data that can be translated into evidence. And I just wanted to mention the regulatory landscape here in terms of the US and EU. There's a lot of development in here right now. And I think we all as a field also really need to watch that and grow and learn together as more guidances are coming out. At the forum, we've had a series of webinars on translating real world data into real world evidence. I've put the link here where we've covered some of these things. So this is just from the most current website at the FDA in terms of all of the new guidances that are coming out. And just wanted to make sure that these are up here. And then in the EU also, they have a fairly large program in real world evidence and how it is used in EU medicines regulation. And this is from a recent review by Peter Arlett and Emma Cook, who is now the director of EMA. And this includes 11 separate work streams that were actually described in much more detail at that meeting that we had, the webinars that I referred to earlier. So both US, what I'm trying to say here, both US and EU have a lot of activities going on right now, and there's a lot of development, and I think we all need to watch this space. But how do we then jump from real world data into external controls? Going back again to these meetings, these webinars that we had, this was a presentation by Bettina Hansen, who's one of the statisticians that's so incredibly good in this. And it's really about trial data versus real world data. And she was explaining some of the difficulties and the statistical approaches that need to be taken, but basically making the point that it is feasible to do this, but there needs to be a lot of collaboration and willingness and trust, a lot of data sharing. Protocols are so important. And of course, the statistical analysis plan is paramount. But really, this sort of building this collaboration between clinicians, pharma, regulators to improve patient care. And one of the cohorts that Bettina Hansen has worked on is the GALA, which which I will talk about a little bit later, which focuses on patients with Alagille syndrome. And this is just one example. Livmarli was approved in September 2021 and it's indicated for the treatment of the cholestatic pruritus in patients with Alagille syndrome. And this was based on a randomized withdrawal study design. But what they were able to do with the GALA study group, and this was presented at the ASLD in November of last year as a late breaker abstract, really a very good example of how real world evidence can fit into this. And so basically they used the GALA external control cohort, which was aligned to be aligned and harmonized to the barabixalat cohort. So really you take the real world data, you then align it, And I don't have time to go into all of these details, but it's just a very good example of how this was done. And of course, it involved a lot of, because it's a rare disease, a lot of patients from a lot of different countries. So again, the need for collaboration and harmonization. But you can see here that when you start off with all the real world data that GALA has, by the time you have your harmonized control groups, it's really a very selected group from that. And all of the different steps are outlined here. And so they were able to show them that, very visible in this graph difference in terms of time. So basically event-free survival between the treated cohort and the GALA control. And this obviously really lends a lot of confidence to the use of this drug and its impact and benefit for patients. Here again, many sensitivity analyses that were done. And basically, I'm just kind of showing this as one example of a very well done real world data evidence generation to be used as an external control. Now, Rashmi had asked me to talk about this paper that we published, which is just basically a review of external controls that were involved in FDA regulatory decision making. It's just meant to be a review of when real world data has been used and how it was used. And again, we are talking about external controls, which can be concurrent or non-concurrent. And these non-concurrent basically then are historical. So the data was collected in the past, but it can be retrospectively collected data or prospectively collected data, or it can be published data or from previous clinical studies, or sometimes also baseline controlled studies. So the type of data and how it was collected becomes so important. And when we look at the FDA guidance and also the International Council Harmonization E10 document is, really, and I think this makes a lot of sense. The use of external control design is most persuasive when, obviously, when it's not possible or ethical to run a placebo control, and there's no available therapy for comparison. The disease progression is well understood and predictable. I think some of these are so important to have those well established. Also, that the outcome measure is objective, and that there's if there's a dramatic treatment effect, as we saw in the marabixalat example, that again is so important to generate confidence in the treatment effect that we have seen. And again, these can all be reviewed independently. I don't have time to go into each of these, but it gives you basically, it gives us an idea of when are these historic controls or external controls most useful or most convincing. So in this paper that we published, we found 45 studies that were supported by external controls. The majority of these were for rare diseases. Retrospective, historical, and retrospective data was the most common source, and objective endpoints were obviously very important as primary endpoints. So here we see that the majority of them, a lot of them were gastroenterology and inborn errors, non-malignant hematology, metabolism, endocrinology, etc. And here again, we can see the kinds of natural history studies that were used, whether it was published data, baseline control, but the majority of them were retrospective or historic natural history studies. What also is discussed in this paper, and again, I won't have time to go into all the details in this short talk, but having the right statistical approaches to reduce bias is so important. And again, in those studies that Bettina Hansen presented based on the GALA cohort, natural history cohort for Alagille syndrome, it really illustrates the different statistical approaches that are necessary and to really structure this external control. And I just wanted to give one example from the paper for Zolgensma, which was approved on May 24, 2019. This is one of the examples for rare diseases. It's for SMA. And it was approved based on a completed phase 1 dose escalating study with 15 patients in it, and an ongoing phase 3 single arm study with, I believe it was 21 patients in there. And the comparator was a 2014, it was a study where we have a publication from 2014, which was a natural history study where they followed children without treatment, without pharmacological treatment with SMA. And that really provided the basis of comparison for the treatment effect seen here. And in this publication in 2017 in the New England Journal of Medicine, we see here that this is the dose escalating study with 15 patients. And what we can see here is the cohort 1, they received a lower dose, and this is the time of the infusion here, and this is the end of the study. And this big circle here means that there was a pulmonary event. So the first 3 patients received the low dose. The next set of patients, the other 12 patients, received a higher dose. And as you can see, there was no pulmonary events here, but all of them surpassed this 20-month line, which basically in the natural history cohort, only 8% of the kids had actually surpassed that. So as it says here, the thicker vertical line indicates the benchmark of 20 months, at which time only 8% of the patients with the disease typically survive without permanent ventilation. So it's based on that 2014 study, but also a lot of clinical experience with this disease so that we can be, it generates a lot of confidence to see that every single one really was able to bypass this. Here we see the CHOP, which is the outcome measure. The Children's Hospital of Philadelphia Infant Test of Neuromuscular Disorder is what CHOP stands for. And here again, you can see the children that just got the low dose and then the children that got the higher dose. And you can see this dramatic treatment effect in terms of their neuromuscular function. And a lot of it had to do with being able to sit up on their own. So I think this illustrates the kind of data that inspires confidence or generates confidence amongst everyone involved, the parents, obviously, the caregivers, the clinical community, pharmaceutical companies to say, yes, we have a treatment effect here, we can build on that. But most importantly, in this context, also the regulators to say there definitely is a treatment effect here. So the key take-home messages here were that we have a lot of examples of external controls being used to support regulatory decision-making. It's not unusual. It is difficult to set up a prospective cohort. It's not impossible, but it is more difficult. And so when retrospective data are available, that is we can find good ways of using that, but it has to be done right. The rigor of the data collection is very, very important to have the appropriate statistical analysis and include a statistical team from the very beginning is key. We need clarity on the course of the disease. Common data standards are extremely important. And then also we need to think about the totality of the evidence and experience we've had in this field. And how do we move forward from here? I think it's so important to then also look at what happens after approval for conditions based on external controls. And I know both in the US and EMA, the EU, in the EU are doing reviews of studies where external controls have been used and how they were used and what the implications have been. But I think it's also going to be very important to then have follow-up, and we can migrate from natural history studies to on-treatment observational studies to really continue looking at the impact of this treatment and to then serve as a control for the next generation treatment for that particular disease. And I think most of all, it's so important that we all learn together. That's really the only way that we can move forward here. And I think for a disease like this one, JIS, it's so good that you're all coming together at this point, because I think you can really start talking about common data elements and data that you can gather in clinical settings now to set up a mechanism to look at data, both retrospectively and possibly also prospectively, as the clinical community comes together to put those structures and collaborations in place so that when you start your studies, that you really have a way of having that control information already available. But most importantly, it's a matter of all learning together. And I thank you for your attention. And I will stop sharing my screen.
Thank you so much. I think I see a question there. Yuki is asking, why are prospectively collected controls so difficult to do?
Okay, that's a very good question. It just means that there has to be the infrastructure in place to do that, right? I mean, when you do retrospective data searches, you have your patient charts, you have your electronic health records, and you can go back and pull data out. But there's not going to be complete data sets. There's going to be a lot of missing data. There's going to be a lot of lack of standardization on how data was collected at the different clinics across the US and across the world. To do it prospectively, you need to come together ahead of time, and the community of clinical researchers needs to agree, okay, this is how we're going to collect the data, and have some standardization in there. So it complicates the normal clinical care a little bit. And maybe some of the clinicians here really want to comment on that. But it basically means you have to commit to something. It needs to be funded, and it needs to be followed in a very standardized, high quality sort of way. Does that make sense?
Yeah, that makes sense. I think Fabrizio has a question or comment.
It makes a lot of sense, Professor Mill. Thank you for your presentation. I'm one of the clinicians who's supposed to try to put this together, and I think you have been very informative. I may bother you and ping you a few times by email. If you
Okay.
Excellent presentation. The point of the perspective is that it takes a lot of time, Rashmi. It's going to take a lot of time. If you want to collect prospective data on a 3 years disease course of a rare disease, may take 10 years. And this, I don't think we have the luxury, as I mentioned it before, right? So retrospective, it's very efficient, time efficient, because, once the point is collecting, and I'm taking an example from the New England Journal of Medicine paper that you showed. They have a very clear-cut, simple outcome. Yeah. That's the problem,
right.
Okay? That is time you get into, you need ventilation.
Right.
That was very hard outcome. So the challenge for us is to define the data we want to collect with something that we think is going to be the potential primary outcome in the future trials, and make it as simple as possible. Does this make sense?
Yeah, I don't know if you're asking me, but
Yes, I'm asking you. I'm asking you.
No,
It looks to me like you are
absolutely.
the expert around here.
Right. And with this disease, which I'm not that familiar with, JIS, there's systemic, right? Juvenile idiopathic arthritis, but then also with these acute episodes of MAS, how do we integrate that? And of course, those acute episodes can be studied differently, and they have a particular length of time that they would last, right? Whereas the whole disease course itself of the systemic disease is a long-term progression. So maybe really kind of figuring out how those two interact and fit together would be important. But that's why I think I was so intrigued when Rashmi invited me here, and I learned a little bit more about this disease, how complex it is, and difficult to really get a handle on it.
Sure.
Can I ask you this? The studies that you mentioned, it looks like you were referring already published data as a sort of as a control, correct? If we go, unfortunately in our field, I don't think we have an existing good quality data set that we could use an external control. Would it be acceptable if we went back and do a retrospective chart review? And again, we would come up with very specific criteria how to select those patients for review. We would come up with the sort of necessary items that we need to collect, but sort of not having this data set at the moment and getting a retrospective review now, when we know what type of trials we would like to do. Sort of, what your take on this?
And particularly, if I may elaborate on what Alex is asking, what are the regulatory implications of going retrospectively now, while the trial is being either designed or even, let's assume, run?
Right, and we do have regulators on the panel here, and I really cannot speak on behalf of them, but I think it all comes down to the quality of the data and the consistency of what is collected. But in the end, it's going to be case by case conversation with the regulators. And I don't know if Rashmi, is it permissible for me to refer that question to Carrie Jolie?
Yeah, absolutely. Yeah, absolutely.
Yeah. Hi, thanks so much. And again, I haven't been here yet, so I have to start with my disclaimer that my views are my own and I'm not purported to speak for the agency. However, so I think that everything is a case-by-case basis, certainly, and that discussion would need to be had, with the division. However, in a general sense, there are challenges with retrospective studies. And the reason I think we haven't seen as many prospective ones is we're still sort of, historical controls have been used for a while, but we're sort of new in this real-world data prospectively collected era. And so now we're starting to see those pop up more, and really people used to just come with what they had. But the issues with retrospective data, you really can run into a lot of selection bias with retrospective data. You generally don't know sometimes whether or not the patient characteristics were assessed that you were currently trying to study in the trial. They may have been assessed differently based on historical approaches. So not only what are the outcomes that you're trying to assess, but how did you assess them? Perhaps instruments have evolved, you weren't using the same types of techniques. And you can have a lot of issues matching patients, right? So comparability between disease definitions, diagnostic techniques to even diagnose rare conditions particularly may have changed in recent eras. And aspects of how they were even followed may have changed. They may have had more follow-up now than they did previously. Aspects of standard of care may have changed, which can make things look very different when you're looking at interventions. And as well as retrospectively, you were not in charge of how the data was collected, right? So when it comes to using these types of retrospective controls, we really still have to be able to ensure the data is valid, that it is complete, and sort of that whole data provenance is as rigorous as we need it to be. So there are a lot of challenges in using retrospective studies.
Right.
I see also Nikolai. Do you want to comment, Nikolai?
Well, I wouldn't dare to comment anything more than what Carrie Jo did, because she is certainly more experienced in that space, and she has more experience than I do. But I think this came also in one of the comments in the previous sessions, that to rely on external historical control, we need to think about whether patients in the historical control would be reflective of the patients that we see now, given that standard of care has changed, particularly for SJ as a disease where we have drugs approved now. And the question is whether the patients from the historical control would be representative of the patients that you want to study in this open label or whatever control therapeutic arm, and whether we have sufficient understanding to know that, say, a certain endpoint would behave the same, or a certain outcome would behave the same now than it did, a while back, 5-10 years ago. So I think these are important considerations to think about. And then the rest of the points that Carrie Jo and Veronica very nicely outlined, I think they are all important.
So it specifically for us would mean that we have to study. We have to, if we go retrospectively, which I think we have to, for the sake of time, we have to include patients that are in the biologic era, at least after 2010. Or 20, whatever. Okay, and maybe one of the inclusion criteria, because I've tried to put together some high level things for such a registry, and maybe one of the things we should include is failure to respond to. I'm thinking aloud then.
Yeah, it's Carrie Jo. If I could just add another point, a guiding principle in the real world data, real world evidence space that I often like to espouse is, can your data answer the question you are asking? And so really what I would encourage you to do is think more specifically about what the question is that you are trying to answer is, and then look at the data set and see, do we have the right inclusion exclusion criteria in the data set? Do we have the correct patients? Do we have the correct outcomes? Do we have the correct measurement instruments? And that's really a good way to go about sort of designing any type of or utilizing any type of external data, external control data or real world data.
Right, and sometimes as imperfect as it may be, going back to collecting the data that you currently have available retrospectively, maybe setting some time boundaries, maybe, you won't go back all the way to 1960, but maybe 2010 or less than that, as you mentioned, the biologics era would be good. But so then you at least have a baseline of the data that you have. And maybe that could also serve as hypothesis generating. And then you can really design your studies to test some of those questions as you move forward. But I think as a field, to me, it just seems to make sense that you would at least gather what you currently have. And, what are you starting off with in terms of data? Because that can then really inform how you can then set up a standardized prospectively collected data. Would that make sense, Carrie?
Yeah, no, in fact, we often use those types of real world data, whether they're natural history studies or registry data, to inform trial design.
Right.
And that is one of the strongest uses of that type.
Right,
Yeah.
right, yeah.
I'm going to be talking about the CARA registry in a few minutes. Actually, I think that we have been collecting data on multiple, pediatric rheumatic diseases since 2015 in a pretty rigorous and standardized way, prospectively, and as well as in some cases retrospectively. So I think it would be a great idea to really look at the data that is being collected there to see if, some of the patients enrolled who have systemic JIA could meet the criteria that might be used for a study and be the controls for those patients, or even also recruit them at the same time to be, contemporary controls at the same time as the clinical trial, potentially. Is that a possibility?
Are you asking everybody or?
You?
Me?
Yes,
Yeah,
Karen.
no, I mean, these are new methods. So I will say we are still learning. I mean, is that a possibility? As long as we can design something that is adequate and well controlled, augmentation, should we be able to really work around matching and inform inclusion, exclusion. And, the other issue we run into with real world data is for patients who are not actually in the trial is what is your time zero, right? Which essentially means from what time are you measuring the effect? So if they're not in a trial, when does that time start, right? And a lot of that depends on how well defined also the natural history is of the condition you're looking at, right? So is it, you can really project that everyone progresses from A to B over X amount of time, trajectory, or is it very heterogeneous and we're still not sure about whether or not there's subsets and who's going to, reach which milestones when? So all of those things are considerations that really need to go into that, that ultimate determination of how much those patients can be used and in what way.
I guess one question I have for the field is, this is a juvenile disease, right? What happens, because we've come across this in some of our other rare diseases, what happens as they grow older and move from the pediatric setting into the adult setting? Because that can often be a challenging time as well for data, because they kind of get lost in the transition from pediatrics to adult care.
This is a real challenge, but some of these refractory SJIA with this severe systemic predominant picture and lung disease and relapsing acute macrophage activation syndrome don't make it to adulthood.
Right.
Right. And this is a phenotype that we have been learning to recognize, and which appears to be particularly frequent in the US. And Alexi is an expert rather than in Europe, but is rare. Okay, so we really don't know what it is. That's why we really think that we need to do something for these patients. We have becoming more and more aware about the pathogenesis. We understood a few things, and we have potential, drugs and companies interested in the field. So this is important.